Okay. Hello, everyone. Good evening. Welcome to the 7th session of data analysis for biologists live session. So I'll just start sharing my screen. In the meantime, do you have any questions, any queries about any of the classes we've had until now? I would also like to mention right now that we'll have a quick revision session on Friday. That is the 24th of March, same time, 6 p.m. on the same link. If you have any questions about your exam, which is on Sunday, you can ask. I'm planning to have a quick recap of the major points from all the classes, and we'll be solving some of the important assignment questions. I don't think we'll have time to solve all of them, but just the important ones or the more difficult ones. And you can ask your queries in that class. All right. Okay. So let me share my screen. Okay, I hope you can see my screen now. Yeah. All right, great. So I think we can start the session. Okay, so fine. So in this, okay, so we've had, yeah, so here this would be 7. So we've discussed 7 weeks of the 8-week course until now. And so you've seen all those videos, and we've had these live sessions where we have solved questions. So all the recordings, they have been uploaded to YouTube. The scripts that Sir showed in these lectures and that we have used here in the live sessions, they were all been uploaded to Google Drives. I hope you have the links to all of them. You can use these codes as it is to practice. Receive week 5 lecture solutions. Sorry. Could you just unmute yourself and let me know which one are you talking about?
I think after week 2, we didn't receive the solution for the lecture questions, whatever Sir has asked during the lectures.
In week 5?
Yes, week 5, from week 5.
From week 5. Okay, I will check and get back to you.
Sure, thank you.
Were there only 1 or 2 questions in that week? Or were there more?
I think it's 1 or 2.
Okay, fine. Either way, I'll send you the solutions as soon as possible.
Sure, thank you.
All right, fine. Okay. So, all right. So now we have come to the last week of this course, week 8, where we're talking about higher dimensional data and how to work with such data. So basically, it's known as reducing dimensions or dimensionality reduction. And again, just reiterating that in your exam, which is on which is on Sunday, there are going to be no questions that require you to type in on RStudio or that require you to do complex matrix calculations on a tool. But the basic funda that you have understood through these lectures and simple calculations, those will come up in your exam, right? So they'll be testing your knowledge of R and any of the topics that we have been studying until now. And you'll also have that sample online calculator that you also already have the link to. Fine. So today we'll talk about some examples of higher dimensional data in biology. So the first one right here, this is microarray. So what happens here is that there are some probes. Let's say there's, so you may be familiar with the concept of fluorescence. So let's just say that these probes, they have a fluorescent molecule attached to them, and they give out this fluorescence when a complementary strand, a complementary RNA, RNA with the complementary sequence is attached to it. Only then does it show fluorescence, right? So now let's say there are two types of cells you have. You want to compare a healthy cell and let's say some diseased cell. And we understand that when a cell is diseased, it's going to be because there are some differences, some changes, some mutations, maybe at the DNA level, or these may be at the epigenetic level, so that some methylation marks on the DNA or proteins. And these ultimately manifest in the form of changes in RNA. So all the RNA of a cell or multiple cells, that is known as the transcriptome. So here what we are basically interested is, we have so many different RNA, we have so many genes. Each gene will have, so whatever number of genes are expressed in that cell, all of them will have their own respective RNAs. But now in a diseased condition, as compared to a healthy control condition, in the disease state, a few RNA, a few genes may not be expressed at all, or they may be expressed while they were not expressed in the other case. So to compare such a situation, what is done is, so here it shows an example of microarray, where you can see that the one which is gray in color, that means RNA from neither of the two conditions was present. RNA in neither of the two conditions were present. And the ones in green, it means that only the normal cells had this RNA molecule. The red ones here indicate that only the diseased cells had this RNA molecule. And the ones yellow, they mean that the RNA molecule was present in both of them. So the gray ones and the yellow ones are not really of our interest, because the state is common across our two conditions. But the red and green ones are more informative to us. So now in this case, it is not always that you'll be working with just, so here you can see that there are so many, so each well here indicates a different RNA molecule. So that's like a feature that you are talking about. This is something which is differing across multiple patients that you would be testing through this. So here, you will have patients on one axis. Like let's imagine a table. So you would have different patients on, let's say, as rows, and each of these RNA molecules or the genes in the other, let's say, in the columns. So that becomes a very high dimensional data. So you would like to know how you have many healthy patients and many like healthy controls and many patients, and you would like to compare across the two. Similar is the case of single cell RNA sequencing. So now imagine, instead of comparing several patients and comparing expression profiles in several patients, you have just tissue sample from one patient, but you are able to segregate every cell. So let's say you have taken 5,000 cells at a time, and you are able to separate. Imagine you have a well which has a plate which has 5,000 micro wells in it, and you have been able to put one cell without bursting open the cell. You've been able to put one cell in each of these wells, and now you are performing sequencing on this. So what do we mean by sequencing? That now you are able to identify how many RNA molecules are present from each gene in each cell, and to what extent, how many molecules are there. So typically these values are expressed in counts per million. So let's say if the total number of transcripts, total number of RNA molecules in a cell is 1 million, how many came from gene 1, how many came from gene 2, how many came from gene 3, and so on. So here also, now let's say 5,000, these are your data points. Let's say number of rows, and your columns, when you're talking about number of genes. So it could be, so here there's no really limit on how many genes you're testing. You're testing all the genes. This is not targeted like microarray. Whatever is expressed is there. So let's say 25,000 genes on an average are expressed in that tissue. So we have that information. Now, what would you like to do with that information? First of all, you would like to identify what are the cell types present. And when we say cell types, they could be cell types in terms of just different types of cell. Let's say if this was a liver tissue, so you'll have hepatocytes, Kupffer cells, and many more. Another way of defining cell type could be cell state. If you're able to distinguish the different, let's say, you're able to define that, okay, so many cells are in M phase, in G1 phase, and even in mitosis, you're able to differentiate into prophase, metaphase, and so on. So this sort of information can be gathered from such a data. Another example, which was also mentioned in the lecture, was high throughput microscopy. So you have a lot of cells at any time in your screen, and you are taking these pictures at some interval. So there are so many parameters you would like to determine from that data. So what is the average not just average, but like for every cell, you would like to know how much is it moving. That's like the displacement. Is the size changing? Is the area or like the circumference, whichever parameter we would like to know. This can also be accompanied by some gene expression studies on the other hand. But nevertheless, the high throughput microscopy itself is a high dimensional, it gives a high dimensional data as output. Another example which you can see here is, so whenever a patient's blood work is done, you have the full blood count, you have so many parameters identified, right? So this can be, each of these, this becomes a feature, and this can be again compared across people who are healthy and people who suffer from some or the other disease. And then it can be compared that which are the features which are most informative, or a combination of features that are most informative in distinguishing between these two groups. So typically, if you would find out that what finally we want to do with such data is to make some groups or find out differences across these groups. So now the issue with such a high dimensional data is there are two main challenges. One is to visualize, because we would like to see them and infer from it. And normally we would like to have it in 2-dimension or a maximum of 3-dimension, because even if we have 3D data, we can make 3 different plots, like so dimension 1 and 2, 2 and 3, 1 and 3. And then finally also analyzing this data. So the best option is to actually club these existing dimensions into a number of lower dimensions. So let's say we are starting with 20,000, we bring them down to 10 or 50. So there are many algorithms that you can use to analyze data in 30, 40 or 50 dimensions. And typically what happens is that the first 2-3 dimensions are the most informative. And these are the dimensions that we can use to visualize our data also. So here is an example. Let's say we have these 6 points in our data. So one way of reducing dimensions is to just select one of the existing ones. So we have 2 dimensions to begin with. We want to reduce dimension. So the answer is we need 1 dimension. Now, should it be this x-axis right here? If we project each of the data on the x-axis, you can see here that when you do that, out of the 6 data, you will only get 3 unique data points. So you have lost half the information. So let's go to the other axis. We project these data on the y-axis. So here also you can see that, okay, out of 6, we are able to get 5 unique points, which is not bad. We have sort of retained most of the information, but we would still like, if all the 6 data points, if we can retain all of them. So what can be a solution? Let's make a new dimension. So if you fit this dimension, you can see that each of the data points will have a unique projection on this, and the heterogeneity is retained. Now, how was this whole thing done? So one of the ways to do it is PCA, or principal component analysis, which is a linear dimension reduction technique. So what we do here is, so now initially you begin with D dimensions here, and you identify another D dimension space. You start with 20,000, you identify new 20,000 dimensions. These are also orthogonal to each other. Now, the initial ones need not be orthogonal, right? Because let's say these were genes. So we don't know if these genes are orthogonal to each other. But the new ones that you identify, each of those 20,000 are going to be orthogonal to each other. And they have been determined in such a way that you can actually see that some of them are more informative than the others, while most of them would have negligible variance. So they are not at all informative. So now you select the top most informative of these dimensions, and project your data on that, and that becomes your new data set to visualize as well as analyze. Now we take 2 examples. First of all, we start with a 2-dimensional example, so where we project it on 1 dimension, and then we begin with the D dimension data and we just generalize it. So let's say we have 6 data points here, x1, x2, x3, x4, each of these, and the dimension D is 2 here in this case. So it's like an example of your XY data space. So a new vector, we call it U. It's going to be unit vector because so a dimension, an axis, it doesn't make sense to have a length to it, right? Your x-axis can go from anywhere to anywhere. It does not have a finite length. So the most important part about the x-axis is its direction. So we identify a unit vector. It means its norm is 1. Its direction is what we are more interested in. So let's call it PQ. So now the important aspect is that this is also going to have the same, like, so ideally we say that it's going to be a combination of the existing dimensions. So it will also have two, like, number of elements in this dimension is also going to be the same as the starting D. Now, it is possible that one of these may be 0. So when we take the other example, it's possible that some of these elements in U may be 0. So that we'll see later. So what we do is that we project each of our data points on this new axis. So let's take the example of x1, y1. So if U is PQ, the output is here, it's going to be the dot product between x1 and U. So it becomes px1 plus qy1. That means the original information, which is x1 and y1, that is retained. It's just changed by a proportion p and q respectively. So it's a linear combination. Now, an important aspect of performing this thing is, first of all, when we start with our data, we must center the data. Centering data means you find the mean and you subtract the mean from each of your points. So now the centered data will have a mean of 0, right? You do it for all the axes. So the first axis x and the second axis y, and then you find a covariance matrix. This becomes your covariance matrix right here. Yeah, so now we'll begin with this covariance matrix in the second example. Likewise, when you have a d-dimension space, so here it's just important for you to remember that here we have these dimensions or these features in the as columns, and each of your points here is in the rows. So this becomes your initial input, and so now you would like to find many such unit vectors. Each of the unit vectors will have this feature. So and here I'm not mentioning that x is, like, I'm not centering the data because I'm assuming that we have centered data to begin with. So this is important. If we do not have centered data, then we mention that, okay, center the data and then proceed. So here is what we're going to do. So we'll project the data, project our X matrix on U. So it becomes XU. This becomes the new format. Here also we are going to have the same covariance matrix. Just a second, yeah. The recording is going to be uploaded on YouTube, all right? Okay. So here it's the covariance matrix. Now, what do we do? So once we have the covariance matrix with it, with us, so now this is a square matrix. And yeah, now we would have some eigenvalues and eigenvectors, right? So you remember the formula, right? Matrix multiplied by a vector gives a scalar multiplied by that vector. So that vector is going to be called an eigenvector of that matrix. In this case, we are talking about the covariance matrix, and these corresponding scalars are going to be our eigenvalues, lambda. Now, the idea is that if n is greater than d, that means you have more points than the number of dimensions, and each of these dimensions are linearly independent to each other. So you would have a maximum of d eigenvectors. This is something we had also learned about when we were talking about eigenvectors and eigenvalues. And if n is less than d, then at least one of these eigenvalues are going to be 0. So that is something we already understand. So what we do with these eigenvalues and eigenvectors is, so the main idea is, so we began by saying that, okay, once we have identified, we started with d dimensions, we'll get, let's say we get d more dimensions, right? D of these eigenvectors. So we want to somehow bring down the dimensions now. And we want to choose only those dimensions which have the highest variance. So here it has been shown. So what we want to do is we want to maximize the variance of XU, because that is our projection. That is the data we are projecting. So it has been shown that this is equal to the eigenvector transpose multiplied by that covariance matrix and multiplied by the eigenvector again. And we also know that SU here is going to be lambda U. So it becomes UT lambda U. Scalar can be moved before, and U transpose U becomes the identity matrix. So finally, we would just have lambda with us, right? So that means variance of a matrix, once it's projected on a vector, which is actually the eigenvector of the covariance matrix, is equal to the corresponding eigenvalue. Now, that means, so you had d dimensions to begin with. Now you have d, again, you have d dimensions. Each of these are orthogonal. Corresponding to each of these dimensions or these eigenvectors, you have eigenvalues. But now what you can do, you can arrange your eigenvalues in the descending order. So lambda 1 is going to be the greatest, followed by lambda 2, lambda 3, until lambda d. And you can only choose some of the first few lambdas because they are explaining the most variance of your projected data. And these first few eigenvectors are going to be the principal components. So the name suggests that these are the components. So all d of them were explaining your data. Now you took a few components of them which explained the most, or they were principal in explaining your variance, and we discard the rest. So now we have, so this is the idea, but how many should we take? So what is done normally is that, so each of them are explaining a proportion. So let's say, so each of them have a certain value. Now you sum them up and you divide each of the lambdas by that fraction. So what it means is it's going to tell you the total proportion of variation that is explained by each of these principal components. And these you can plot like a bar plot or a scatter plot. And here it's known as a scree plot. So this is what it looks like. You had initial data, n rows. Just a second. Right, so the dimension here, we mentioned rows first. So n times d, right? And now you have the loading matrix. Here, each of the loading matrix has n elements in it. Loading as in your eigenvectors here, each of them had d elements, right? So d, and how many such eigenvectors? You had d of them initially, but you chose only a certain number of them. So you chose, let's say, m of them. And so now by matrix multiplication, you would have m times n times m. So these are the names. You call this the first one, where you are actually, this one is actually telling you the proportion of each of these dimensions. That becomes your loading matrix. And then the final value, the final projected data, that is your final score, right? And each of these columns here is your principal component. So let's say normally we only use the first 2 or 3 principal components to plot. So you would only plot this and this. Normally, the first principal component is plotted on the x-axis and this is plotted on the y-axis. All right. Yeah, so here we are at the end of principal component. We'll move on to RStudio now and take a look at a few examples. In the meantime, please ask any questions if you have. This is a complicated topic, dimension reduction. So please ask if you have any doubts. All right. Okay. So before we actually see a few examples, I would like to mention just something. This is not exactly a part of the course, but it is worthwhile to mention. So let's see these 2 examples right here. So I already have them loaded because it takes a little while to load. So we talked about single cell RNA-seq data, right? So I work on it. So I just wanted to show you this thing. So this is what this one looks like. So DM here, I actually have that matrix. So you can see I've clicked on this and it's taking a while to open. So normally, so if you can see here, this has, as far as I remember, some 20 or 30,000 rows, which are these genes, and 5,000 columns, which are 5,000 cells. So it takes a lot of time to open. You can see this has 155 million elements, and this is taking 1.2 GB of space, right? So this is what it looks like. So if you are interested, you can take a look at how each of these cells is individually marked, because it doesn't make like it's not very cost efficient to sequence each cell individually. So somehow all the RNA molecules from every cell, they are attached to a unique DNA or like a nucleotide barcode. It's known as there's like the cell barcode. So And these are the cell barcodes you can. So each of these columns is a cell which has a unique barcode to it, which is a combination of ATGC. And these things that you see here, these are your names of genes. Now, can anybody tell me something which they feel is a little different than the other such count matrices that you might have seen through the course of these lectures in this topic? Right? This is a count matrix, right? So normally a count matrix has genes as rows and samples or cells as columns, but it may change. Okay, different types of input can be required by different algorithms. So it's not exactly a hard and fast rule. But can somebody tell me what is something different about this count matrix, or something that you find is a little strange? Okay, so if you don't find anything strange, there are a lot of zeros that you see here, right? And you can see that this is irrespective of which column and which row I take, you'll always find there are a lot of zeros throughout. So what happens is when you are working with single cell data, there's a lot of information that's missing. And this means that from every cell, you are actually not able to identify every gene. Only the most expressed genes are mostly captured. So what happens is you have so many zeros in this. And this is why, anyway, so you have whatever n times d number of elements here, which is 155 million. And there's some 1.2 GB of information here. So what has been done to sort of reduce the size of such a data is that it has been converted into something that is known as a sparse matrix. We already know what a matrix is. So this was an example of a matrix because we just had numbers all throughout. So here we have a sparse matrix. Now, you can't exactly view it here. So I would just like to do a head command here, and then we can see. So now you can see here, this is the same thing. You have rows as your genes, and these are your columns. But now instead of zeros, you have dots, just a dot, right? And all the non-zero numbers are there. So now you can see the size has reduced tremendously. It's almost 6 times less. So just a side comment. So when you're working with high dimensional data, higher memory is also a problem in that. Fine. So now let's come on to PCA. So this was, so for this, I'll give you an example of the iris data set that I think we're all now very familiar with. And we'll be plotting it using the same base command plot, as well as using ggplot because this has some additional functionality. So to install ggplot, you need, so normally what we do is if you install it from here, tools, right? Install packages, you would see that what actually happens here is the command install.packages. And in bracket and in double quotes, you write the name of that package. Now, this one is not directly available via this method. So you need to do a slightly different method, which is install_GitHub. And then in bracket, you write the name of that package and where it is from. And for the install_GitHub command to work, you need to do something else, which is this would be install.packages DevTools. So first you install DevTools, you call that library. Then within that DevTools package is the option to install via GitHub. And then once you've installed ggbyplot, then you can call that library and work. All right. So let's start with the iris data set. And as we all know that the first 4 columns here are numerical data, and the last ones are actually the species. So what we'll do is we'll take the first 4 columns here as our I data, right? So 150 observations of 4 variables. And so the prcom function, which is available in base R, we feed this thing to it. And we say that, okay, you center the data also, that is you subtract the mean from it. And you also scale the data. And scaling the data means making the variance 1. So once it's done scaling and centering, it will compute the principal components. And so now we are going to look at the summary. So before looking at the summary, actually, let's take a look at this. So now if you see here, these values that you see, standard deviation, these 4 values for each of the PCs, this is there, right? These values that you see. However, the other two, the proportion of variance and the cumulative proportion, is actually not present in your data. So while the summary function can give you this, we are not actually able to have it, like it's not present in the i.pca data. So which is why when we want to make that scree plot, we would actually need to compute it ourselves. So another important thing just to note that always you would have as many PCs initially as the number of dimensions in your data. So we began with 4 dimensions and we have 4 PCs. But now we will see how many of these PCs are actually informative for us. Any question, anything? Everything okay? All right, okay. So what we do is we want to find these values actually. So what are these values? These are, as you remember from the PPT, that the standard deviation, square of it. So we have the variance and we want to divide it by the total variance. So this gives us the total variance, which is going to be our denominator. And then we're just going to, so now you remember the variance was 1 for each of the columns. So the total variance is 4. And now we find the variance for each of the, so this is something we already know. And now we are just going to compute this. So if you see PV here, this is the same thing as this, right? And now we just plot it using the simple plot function. So now just to note here that normally what we do in the plot function, we give it the x coordinates and the y coordinates, right? But here we have not given it x and y. We just gave it one list. So what it does is it assumes that this is going to be on our y-axis and the x-axis is going to be as many elements on the list. So 1, 2, 3, 4. That becomes our x-axis. You can also explicitly mention the same. The plot will remain unchanged. And we can give it the x label and the y label. And we have plotted this. That's one way to plot it. So here you can actually see that around 0.7, which is 70% of the variance, is explained by BC1. Also, obviously, we also have this information here. And around 20% is explained by BC2. And the others have only 3 or 4% and not even 1% in this. So we can actually proceed with 2 of the PCs for anything else. So now our initial dimensions were 4. So we actually don't need the third and fourth PC. The first and second PC are able to actually capture almost all the variance in our data. But let's just also look at the cumulative variance here. So the sum plot, when you give it this, it's able to sort of give you, so what the CPV does is, the first one remains unchanged. The second element is going to be first plus second. Third element is going to be 1 plus 2 plus 3. And the last is going to be the sum of all of them. And that's going to be 1. So this is there. And we can also plot this. So it's going to be the shape. And here also we can see that the last 2 points reach quite close to 1. So therefore we don't actually need this one. This is able to explain more around, around 96% of our variance. This is good enough. Now, the 2 information that we saw there, one was the loading matrix and the other was the scores or the scoring matrix. So the loading matrix is stored in under this rotation thing. So i.pca dollar rotation. And the other thing, which is X, this is actually a projected data, finally projected data. So we can take a look at both of these, right? So that means 0.5 times the value of sepal length becomes your PC1 plus minus 0.37 times, sorry, let's just look at PC1 for now. So this is 0.52 into sepal length minus 0.26 into sepal width plus 0.58 into sepal length plus 0.56 into sepal width. This combination is PC1, likewise PC2, PC3, PC4. These are all perpendicular to each other, which means orthogonal to each other, but PC1 and 2 have the highest variance. And we can take a look at the scoring matrix. Now this will have the same number of rows as your data, right? So if you see here I.score, this has 150 observations. Number of columns are also initially the same because we did not specify as of now that we are taking just the first 2 PCs. So here I've done view bracket this thing. So view bracket, the object's name is same as clicking here. So let me click this thing here and you see what command comes here. So when I've clicked this, it says view. So we can directly type view, and this is with a capital V here. Okay. So this is, okay, the initial data also was 150 by 4. Here it's also 150 by 4. And now it's up to us that we only want to plot the first 2 columns. So that we can do simple plot function. We give it the first 2 columns. And now we want to color those points. So if you remember in, so what is now so this one, right? Square bracket, comma 5. What is this going to give us? From the I data frame, it's going to give us the 5th column. And what was our 5th column? It was the species name. In the same order, right? The rows are not jumbled up. Now, what I want is I want to assign different colors to each of them. So what thing I could do is I could just give it a list. Color is equal to C, bracket, double quotes red, comma, double quotes blue, comma, double quotes green. But let's say I don't actually know how many species are there or something like that. So what I do is so we remember that this is a list and this is a list of characters. And actually these are levels here. So the best part about levels is, let's say we convert this to integer. So what happens is now it just becomes 1, 2, 3. The first, whatever the first type of character was, it became 1, the next one became 2, and the next one became 3. So even though I've lost the information of what it is, what I have 1, 2, and 3 with me. And when I feed 1, 2, and 3 to colors, it's going to give me the default colors that are available in R. So I think the first one is black, the next is red, and the third one is gray. So anyway, so here we have 3 colors with us. So we can see that PC1 and so PC1 especially, because this is PC1. So here it's able to differentiate the one which is in black, but the red and green one are not as separated out even through PC1 and neither through PC2. A better plot in this case can be the GG by plot. Okay, I need to call the function here. So library, it's loaded. So here, just to mention that whatever you see here is, these are known as dependencies of a package. So we know that ggpyplot is, it comes up from the ggplot function, and internally it needs some few more packages to work. So anyway, we have this ggpyplot. So we feed in this i.pca. We say that plot the first and second dimension, observation and variance scale, they're all 1. And groups, that means I want to group based on the last column, right? The 5th column. So these were my groups and the classic theme. So this becomes, right? So here we can see the original 4 dimensions with us. And this is the same plot as you saw previously. So PC1 is explaining around 73% of the variance, 22.9 is explained by PC2. All right. So that's one example. And yeah, so here I've taken one example from the Rattle package. Okay, it's just as a slightly, just a different example. So you can skim through lots of data. So there are a lot of inbuilt data also, like they are already available by default, and some of them you can install some package and they would have this. So here you can see there's this wine data here. Yeah, this one. So what it has, wait, just a second. Yeah, this one. So here we have lots of parameters about some kinds of wine, and the type here, it's again like these numbers 1, 2, and 3. So there are 3 different kinds of wine and lots of parameters about it. So we do the same thing. We want the non-numerical columns to be removed. So we remove the first column and we perform PC on that data. Same thing, center the data, scale the data, and we look at the summary. So now here, can anybody tell me how much, so what does this data set look like? How many PCs are good enough to explain most of the variance? So here, now the dimensions have increased. Okay, we start with 13 dimensions. So it is not always possible that you would be able to explain all the variance in just 2 dimensions. Okay, so we find out the PV and we plot that. So this becomes our elbow plot, right? So why is this the elbow plot? The scree plot, same thing here. So when this sort of becomes a straight line here, we think that, okay, maybe somewhere around this bend here, maybe 5, 6, or 7 should be good enough for explaining the maximum amount of proportion. And we can also confirm this using the cumulative plot. So here also you can see, okay, maybe around 7 or 8 pieces, they are able to explain 90%, more than 90% of the variance, which is good enough. So we have reduced the dimensions by half. And so the other thing here, so we can calculate the loading matrix and scoring matrix. So that would not be like, we won't be able to visualize this. So I'm skipping through. Now, yeah, so let's just see the first 2 pieces. Okay, so these were our 13 initial columns, initial dimensions, right? Lots of parameters. So now you can see that so PC1 is sort of, so it's a combination of PC1 and PC2 that are able to distinguish the 3 types of wines in this case, right? Because if you would just have PC1 with you, then maybe a lot of red and green points would overlap around this place. And if you just take PC2, then in that case, red and blue are completely overlapping, right? Because the projection of red ones on PC2 and the projection of blue ones on PC2, they are all on this place. So red and blue ones are mostly separated by PC1, and the green one is mostly separated based on PC2, right? You can also, so we know that the first 2 dimensions were not enough to explain. So you can also visualize the 3rd dimension here. So here I just have the 2nd and 3rd. You can check as many of them, but mostly the 1st and 2nd one. And in some cases, the 3rd one would be required to separate out your data points, right? So we're done with both these examples. Anything about PCA? If not, then we'll move on to t-SNE. All right. So we talked about PCA, that's a linear dimension reduction model. So your new dimensions are going to be linear combinations of existing dimensions. Now, t-SNE and its prior version, which was SNE, these are nonlinear dimension reduction techniques. And the main idea behind these was that they are more interested about the local information. So points that are close to each other, they should remain close in the final data. In fact, they should be brought even closer, and points that were far should be pushed away farther. This is the main idea. So what we do is that we have the original data with us, whatever number of points we have. For all pairs of points, we calculate the Euclidean distances. So we know that typically we denote them by dij. Now what we do is, on the original dimension, we calculate another parameter, which is known as the conditional probability, P of probability of I given J. So what it means, so this is a formula, and this is similar to this half of the normal distributions formula, okay, similar to that. And P I given J or J given I, because as such, there's no directionality to it, right? You go from I to J or J to I. In this case, we are not interested in that direction. So we will just sort of add them up and normalize it by 2n. So now there's some parameter, another parameter here is the sigma. It's called perplexity, and it said that this is going to be between 5 and 50 and definitely going to be less than the number of points we have. So let's say if we have 50 points, perplexity cannot be 50. And in fact, it's expected to be much less than n. So we will see in the example today that even when we begin with the, let's say 150 data points and we try to set perplexity at 50, it says that the value is very high. Okay, so the sigma value should be low when you're especially dealing with less number of samples. So we calculate this conditional probability. This is sort of an estimate. Now, what we do is that, so here we had Euclidean distances and we had a corresponding Pij value. Now, what you do is that in the lower dimension, as many lower dimensions you're talking about, you randomly place or you embed the data. So t-SNE is Stochastic Neighbour Embedding. So randomly points are embedded or placed on that axis. Once they are placed, you again calculate the Euclidean distances, and these are known as, let's say we call them Eij, and their corresponding Pij values will now be called Qij. That same formula. So what happens is, if you have placed data in the lower dimension retaining their spatial information, then Pij and Qij would be very close to each other. Ideally, they would be the same. So now what happens, you define this function. So, and that is what we actually want, right? We want to place our data in the lower dimension, just like it was placed in the higher dimension. So we define a function, loss function, and summing it up over all possible I and j values. So I to j, so I and j are two different points and I is not equal to j. So we take all such combinations and we do Pij multiplied by log of Pij by Qij. If Pij and Qij are equal, then log of 1 is 0. So when you sum all such log values, 0 plus 0 plus 0, L is going to be 0. But we don't live in an ideal world. So L is not going to be 0, but it's going to be a value which is very close to 0. Fine. This was for SNE, Stochastic Neighbour Embedding. Now a slight change was done in how Q is calculated. So D remains the same. Pij remains the same. Eij also remains the same. But now Qij is not calculated through the original formula. Now instead it has a t-distribution like function. And why it is done so? So our aim was that points which are, so the normal distribution, imagine this is the form of P and t-distribution is the form of Q. So let's say if there was a certain, so there was this value right here. So a certain probability P value is, let's say it's this. Now if we, just a second. Yeah. So what happens is that now corresponding to this, so let's take a look at these 2 points first. So let's say if a distance was, this was the distance. Oh, sorry. Yeah. So let's say this was the distance between the 2 points, that is Dij. And if now the distance, this was P corresponding to it. Now, a corresponding Q, the same value of Q, which we actually want, it requires that now in the lower space, the points be shifted from here to here. So they have a lesser difference in between them. So we want to push points that were close. So this is not a very high value. Points that were here, they should be even closer to each other in the lower dimension. And likewise, if we take a point which is here, right? So let's say this one, actually this one. So a point which was here, this one, this D, which had the normal distribution as this one, this was the value of P. Now to have the same value of Q, it should actually be shifted further apart. So this is what we actually want in t-SNE. Points which are near, they should become nearer, and the farther ones are actually pushed farther. And so this is a random process. So you have to set a limit to how many times this needs to be done. So you remember we had also done this in k-means clustering, right? You had a max iter argument to it. So iter, that is one important parameter. Perplexity is another one. We'll take a look at a few more eta and theta in that. And the important aspect is, while you are actually trying to distance away these points which were originally distant in our data set, it's not always perfect. So distances between clusters do not indicate actual relative distances between them, right? Because they were in a very different dimension. So this conclusion should be made, like, let's say you have one cluster here, another cluster here, and another cluster here. So we cannot always conclude. Some cases it might be true, but we cannot always conclude that these two clusters or these two groups of data are more alike to each other than this one. And if you remember in the lecture videos, some of these examples were given. Another thing is that sometimes, let's say, if we have different perplexity values or different parameters while we are performing t-SNE, let's say in one case, this cluster is so big because the data point, they're just spread apart. Let's say we have these 5 data points, but they're spread apart in a different value. It's the same data, but now it's like this. So we cannot say that the data has been, so originally these data points were very different from each other. Therefore, they are clustered like this. They are dispersed. Okay. So this also conclusion cannot be drawn. This is to visualize the data in smaller number of dimensions using a nonlinear dimension reduction technique, t-SNE. And now we would take some examples in R. All right. So the library for this is going to be r-t-SNE. You can again install this using the same function, install.packages. And whenever you're installing packages, you need internet connection. Once you have installed packages, once you don't need anything more from the internet, then yeah, you can switch off the internet and work locally. So we will just load the library r-t-SNE, and we'll also use ggplot2. So like it was mentioned that r-t-SNE has a lot of arguments, and the best way to understand is to try this question mark r-t-SNE. So when we run this, we get this thing, right? So normally only the first 2 dimensions are calculated, but you can increase this. Yeah, perplexity, it can be anything between 50 and 30, the default value. Between 5 and 50, the default value is 30. Right? Then you have theta, which is sort of a balance between speed and accuracy, like that trade-off. So it's also mentioned here that exact t-SNE, it will take time, but you can calculate the exact t-SNE if you have a 0 value of theta. Then you have check duplicates. So this is something we'll see here. So sometimes it may happen that when you have so much data, okay, you have so many dimensions and you have so many points also. So both number of rows and columns are huge. It may happen that sometimes some points will have the exact same value for all the dimensions. That can actually be 0, and that can actually be a different number altogether, but it may not be a unique entry. So 2 rows are actually identical. So it's always good to check that so that you can remove it. That extra point is not adding any information. And then whether or not PCA should be performed first before doing t-SNE, and it's advised that you do PCA first and then t-SNE on that. Then max iter, how many times should this random thing be done? Default is 1000. Verbose basically means that when you are performing it, do you want to get, so a tool does, let's say it has 5 steps internally. Do you want to see each of the 5 steps, something about the step 1 done, step 2 done? So normally it's false because it might be a lot of text, but you can try changing it to verbose is equal to capital T or TRUE in capital, and that you can check what difference it does. What other? So normalize, yes, you should always normalize that data. So by default, it's true. And eta, so eta is sort of the learning rate. Please take a look at what this means. All right. And yeah, I think so. These are our most important parameters, but you can take a look at each of them. So now, in addition to multiple iterations, the first iteration or the first step itself is a random thing. You randomly place values. So Now it's like a random number generator. Okay, you might have come across this in some other if you've taken a computer science related course. So, but anyway, so normally when a random number is generated, it's not entirely random. It starts off with a certain number. So let's say I have performed a step today and I got some nice values. If I perform the same thing again tomorrow, I'll get slightly different value. But now I want to get back the first one. So there would be no way for me to get that value again if I have not set the seed. So I just set the seed. You can set it any value, just keep a note of it. I've set it as 30. We take the same example, the iris data set, right? We remember that the last column was the species, so we remove that. So you can do a -5 or you can do 1 to 4. And right now, let's start doing t-SNE with the default parameters. So what we do is our t-SNE, and in a bracket, we just write our data. Did I not? Okay, so now you see what it says. It says that remove duplicates before running t-SNE. You remember here it was check duplicates, true. So it checked duplicates and it said, okay, there is some duplicated data in the iris data set. Now, what do I do to remove that duplicated data? So you can take a look at this thing. So this command here, it's duplicated bracket and you give it your data frame. So if I run this, it performs, it compares every row with every other row and it shows which of these rows are duplicate. So you can see here, everything else was false except this one. So now I want to, let's say I want to find what is the row number. So I can trace back, but so what I do is I sort of enclose it within. Yeah. Duplicated I dot data. So what it gives me is that the one which was true, it gives me the index. So out of, so in the iris data, I had 150 observations. So the 143rd row is a duplicate of one of the other rows. So I don't need to remove both the occurrences of that duplicate data. I just need to remove the occurrence once. Okay. So what I do is that I'll just put a minus before that and I take all the columns. Okay. So I can just replace the whole thing with 143 actually. So now if you see, so this was initially 150 observations of 5 variables. And now when I run this, it's 149 observations of 5 variables. I also need to change my I dot data, right? So both the things are going to be changed. All right. So now I perform t-SNE again on now this non-duplicated data. And I save it as t-SNE1. So let's try a few more things. Let's try doing it without PCA. Let's try it with PCA, but changing the perplexity from 30 to 5. Perplexity changed from 5, but now, so the default value of theta is 0.9. Now I've changed it to 0.5 to 0.9. And now changing it to 0.1. So these are just random values. So with practice, and depending on what kind of data you have, you change these parameters and then you take a look at each of the outputs, the plot that we are going to make. So for each of them, if you see here, what we do is the output here, let's take a look at one of them, I.tsne1. So there are so many things here. Most of these are just they just mention the parameters that were used, the arguments that were used initially. So the main thing here is going to be y. Now, I.tsne $y, if I see that, so it's in the form of a matrix, right? And 149 by 2 matrix, because you remember we selected 2 dimensions. So t-SNE1, t-SNE2. And but so ggplot needs a data frame instead of a matrix. So we just convert it to a data frame using as.data.frame, and we use that inside ggplot. So, right, so ggplot aesthetics, the x-axis is going to be the first column, y-axis second column, and the colors I want species from my original data frame, which also has 149 observations now, not 50. So keep in mind that this needs to be they need to be in the same, this length has to be the same. And you want a scatter plot, so genome underscore point. Labels here, I want the X label, Y label, the label for the legend, which is, so I gave color here, therefore I'm giving color here also, and the title. So it's the same thing. It's just that whatever parameters I've used, we just want to compare these. So it's plotted. Default parameters, they are actually good enough to compare the three types of flower species here. Y2. Now here what we have done is we have not done PCA. So you can see that they're not as spread out, but they're still able to make these groups. Okay, they're sort of grouping together. But again, remember, this is not a clustering algorithm, so you cannot say that. So here we have just colored them because we already know it. But in reality, maybe this blue one would have been a part of this cluster, and maybe they would not have been called two clusters at all. So anyway, now we take the next example where we change the perplexity to 5. So now you see it's spread out even more. And yeah, so it just has a difference. So you can actually see the t-SNE axis is also changing. The fourth one here, right? So now it's even different. And the last example here. Now this one, so this subpopulation is absolutely separated out, and here the blue and red ones, they come together. So there is no gold standard to what value should be set. You have to try doing this. You have to have trial and error for this, okay? The other example, so this was the same, this was like a cell type data, it's the same as what was explained in the lecture. So here we are talking about two CSV files here. The first one, G, yeah. So again, you can see this is a large data, 366 observations of 16,000 variables. Okay. So now in this case, yeah. So now you can see here, each of your columns are your genes, and the axis here, the rows here are going to be your cells. And correspondingly, so you can see here it's O10.A something. There's a metadata. So metadata means whenever you have some main count matrix, some main file typically against the rows of it, or sometimes the columns of it, you would have a metadata file where it is some extra information, which is not mathematical information mostly, but that is so that information is stored. So like, where was the cell taken from, the age of the cell, or how many times was it grown, or was it the patient's age, where from, so it was brain, which area of the brain, and what. So now they also identified what cell type it was, so they have also given that. Normally this information is not available with us, and we actually want to find out the cell types. So simple thing, t-SNE. So these were some of the parameters that the authors also, so this was sort of what was recommended to get the same results as the authors did. So we do g.t-SNE, we convert the output, the Y matrix, into a data frame, and we plot that. Now we want to color it using the inferred cell type column. So all V1, V2, and inferred cell type, each of these lists, they have 366 entries. All right. So we plot that. Okay, this is taking a while, converting it to a data frame. Oh, this g.t-SNE is taking a while. Okay. So I think, yeah, these are, so we'll, once this is computed, we'll see that you can label each of the points depending on whatever you would want. Okay, so we can take the example of cell types, as you see here. So there were 4 cell types in this data, and you can see that, well, even though we have used t-SNE to perform this dimension reduction, the first 2 t-SNEs are not exactly able to distinguish your groups. And that's okay. t-SNE, the purpose was not to actually differentiate groups, but it was to reduce the dimensions and visualize it in a lower dimension space. So if you see from the metadata, you can use any of these columns as the how to color or label these points. So I've taken this anatomical source. This one, right? Subventricular or ventricular zone it was. So you can see, right? SVZ or VZ. Here also you don't see much of a distinguishing aspect, which is also all right. Another important thing is you can actually color using the gene expression of any of these genes. So any of the column. So since we see here, these are cells of the neurological origin. So we plot one of these important genes, NeuroD6, and the only difference is now we're not going to have absolute two discrete colors, right? Red and blue. We would have a gradient. Expression ranges from 0 to whatever the maximum value is. So what we just say is that, so ggplot has this option that now you color, you have a gradient where the low, whatever is the least amount in that is colored gray, and whatever is the highest value, that's colored red. And once we have that, so now you can see that, so each of these dots, they are these 366 cells. And you can see that, okay, these are the cells right here. They have a higher expression. Most of these do not. Now you can actually take a look at here. So maybe the cells that have this here, they were from the subventricular zone. And actually this one is a better indication. So probably the neurons, the blue ones, they have a higher expression of NeuroD6, right? So a lot of further steps can be, so I think so. I don't have more examples for this at the moment. So, but yeah, so once such a thing is done, now you can identify cell-specific markers. So you would get an idea of which are the cell types there. So you see that, okay, let's say if NeuroD6 is a marker of some cell type. So whichever cells have a certain expression beyond a certain threshold, they can probably be called of that cell type. And then if you have such a data for two different kinds of patients, let's say maybe a patient and a healthy person, then you can compare, okay, these are the genes that have actually changed this cell type across the two conditions and so on. So this is the basis of t-SNE. Now the final topic for today is diffusion maps. Let me know if there's any confusion in PCA or t-SNE. Right? Just a second. Okay. Fine. So until now, when we talked about Euclidean distances and both sort of, especially in case of t-SNE, but in case of PCA also, we did not really keep in mind the geometry of our data. They were data, and you can consider any 2 points, but the distance between point 1 and 2 and 2 and 3, they don't matter, like how you traverse through it. Euclidean distance between 1 and 3 remains as it is. But now in the case of diffusion maps, there's a certain geometry, inherent geometry to your data. And the example, a very good example for this is that Swiss roll data, right? It was like a roll. So actually, let me show you this thing here. So this is one, so if you want, you can try this. So for this, what you'll have to do is first you'll have to install something known as a BioC manager. Again, install.packages bracket double quotes BioC manager, and you install this package through it. Okay, RDR toolbox, and you call it. So there's just this function called the Swiss roll. So let's say I want to have 2000 points which follow this pattern. Sorry, library, and then this. So you see here, this one, this is that Swiss roll data. Okay, here it's just in 3 axes, X, Y, and Z. We are not, for this course, we are not working with this data in R, but we just want to find out the concept of it. So this was a 3D data, but let's say if this data was in higher dimension, how do you reduce dimensions for this? So there we would come to the concept of diffusion maps. So, all right. So now what happens is, so rather than using Euclidean distances, that means if you want to go from 1 to 3, you all of you must have studied physics in 11th and 12th, and one of the earliest concepts was distance versus displacement. So you move from point 1 to 3, you move through this way, or your displacement is like this. So this becomes your Euclidean distance or displacement, and this becomes your actual distance. So here we are actually talking about this distance. Now, this is the distance we are interested in. So now, so this will actually be known as the diffusion distance, but so before we talk about that, if we have any number of points, there's a certain probability that we can go from one point to another point, and it can be the same point again. So if we are at point 1, so let's talk about the first column here, okay? So, or rather, let's talk about the first row. So the first row here, okay? So this probability means that P11, we are at point 1, and we take one step and are still at point 1. That's the probability P11. P12 is from 1 to 2. P13 is from 1 to 3, and P14 is from 1 to 4. So each row indicates, so every row is talking about the starting point. First row means the first point here is the starting point. For the second row, the second point is the starting point, and so on. And each of the columns are indicating where to. The first column is saying that from any point you go to point 1, and so on. And these probabilities, they have this, they are proportional to this ratio, where it's this A minus B is mainly your the Euclidean distance, square of that, and it's to the power. Now, it's a square matrix. You can have eigenvalues and eigenvectors for this. So it has been shown that one of the eigenvectors, actually the highest so one of the eigenvalues, which is the highest one, like in the previous case, we arranged these eigenvectors and eigenvalues in the descending order. Here also, if we do that, the first one will have an eigenvalue of 1, and the others are going to be smaller than that until 0. Okay, so this was the transition probability matrix, where it's a square matrix, n by n, 4 points. So this is 4 by 4, whatever the value of n is. Now, this talked about one step, taking one step from point A to B. Now, if you multiply this matrix to itself once, that is P multiplied by P. Now, each of these entries are actually telling you. So here it's, so like we discussed there, rows are indicating the starting point and column is indicating the end point. So here it's talking about going from point 1 to point 1 in t steps. So here t is equal to 2. So how do you go from point 1 to point 1 in, so in this example, hello, somebody's mic is not mute. Just a second. Yeah. Wait, I'll just share my screen again. Yeah, I hope you can see the screen. So here we are just taking an example of 2 points. So you have point 1 and you have point 2. So so here it's the first row's first element. So that means row 1, column 1. So you want to travel from point 1 and you want to go to point 1 in 2 steps. So the option is point 1 to point 1 in the first step, and again to point 1 in the next step. This one. The other option is from 1 to 2, again 2 to 1. So this one. And you sum these two, because 1 to 1 and then again 1 to 1, or the other option is 1 to 2 and 2 to 1. So you multiply these probabilities and you sum, because or. So simple the rules of probability, right? Multiplication, addition that we talked about initially. And same is the case with all these other elements. So P to the power T matrix is going to give you the sum of probabilities of all possible paths of length T. That is, you have taken T number of steps to go from, so I and J, ith row and jth column, so point I to point J. Now, once you've done this, so let's say now we generalize it. So you've taken some T number of steps here, and we have so many points. Let's say we are specifically interested in finding out. So we started off the problem with finding the diffusion distance. So a different measure rather than Euclidean distance between these 2 points, I and j. So here we talked about that rows are indicating the starting points and columns are indicating the ending points, right? The destination. So let's say we start off from point I or we start from point J. So these rows are what we are interested in, let's say. And now we say any point K. That means any of these columns can be point K, right? This, any of these can be point K. So let's start off with assuming this is our first K value. I hope you've all been able to see my screen till now. I just got a notification, something. Please confirm. Okay, let me just explain it again. So for any matrix, rows are your starting points and columns are the final points in number of steps as indicated by the power on that matrix. All right. So we take two starting points, I and j, and ending point is k. So for time being, let's assume this blue one is your point xi and the red one is point xj. And this column, let's say right now it's k. So now we understand that, so first of all, we only are interested in these two elements. This is going to be some combination of the probabilities. So this is your pi to point k in t steps and pj to k in t steps when this is k. And then you take this as k. So then you are interested in these probabilities. And then so on, each of these are taken as k's. So you sum these up. Now it has been shown, or rather this is the definition of your diffusion distance, square of the diffusion distance from I to j in t steps. Now, another thing is that these, so currently we are talking about these probabilities, right? This one, these elements on their own. Now, what has also been shown that instead of calculating probabilities and summing them up, a slide, just doing some algebra, and it shows that if you actually find out the Euclidean distance between these two vectors. So how do we calculate Euclidean distance? You calculate Euclidean distance between 2 points in any space that you have. So here you have as many, so we remember, right, that this is an n by n matrix, right? So this yi vector and yj vector, each of them have a dimension of n. Each of them have n elements. So if you find the Euclidean distance and you square it up, you are actually finding the same thing as the diffusion distance square. So diffusion distance is both equal to this formula as well as this formula. So this one is slightly simpler. Euclidean distance, we are all familiar with this. All right. So this is Euclidean distance. Now comes the question that we began with, that we want to reduce dimensions. So, so first thing is, how many parameters did we begin with? So initially, let's say our parameters were greater than the number of data points. For example, we had 45 points and we had 1,000 dimensions. So initially, so it's sort of not plotted here, but let's say we have, instead of these 4 points, we had 45 points and each of them had 1,000 dimensions associated with it. Now the dimension of this matrix is n into n, where n is the number of data points. So yi vector that we are talking about, so the ith vector for the ith data point, this y vector has n dimensions. So in this case, 45. Initially, xi had 1,000 dimensions. Now xi has 45 dimensions. So this is our first step of dimension reduction in case of diffusion maps. So now we also saw that matrix P, it's a square matrix. It has eigenvalues and eigenvectors. How many of them? N of them. So let's call them, instead of calling them 1, 2, 3, 4 until n, let's call them 0, 1, 2, 3 until n minus 1. And as we always denote eigenvectors, eigenvalues, normally we start off with the largest one until the smallest one. So we know that the largest one was 1 and the others was smaller than that. So what has been shown is that the first eigenvector corresponding to this lambda 0 is actually 1. It's a constant, right? Whatever that number is. So we don't actually need that for us. Okay. So we remove that now. And now what can be shown is that for the other these psi or the eigenvectors, so that means now you had, let's say in this case, we have 44 of them, 44 of these eigenvectors. Now in the ith eigenvector, we take the ith element. Or rather, sorry, in all the eigenvectors, we take the ith element for the ith data point. And we multiply that in this way. So we take lambda, the eigenvalue, we multiply it with the ith value in that eigenvector, the first one. So lambda 1 multiplied by the ith element of psi 1, and so on. So lambda n minus 1, which was the last eigenvalue, we multiplied with the ith value in the n minus 1th eigenvector. Now this has, so n minus 1 dimensions, right? So your initial xi, it had 1000 dimensions. Then you have yi, which has 45 dimensions. Now you have yi prime, which has 44 dimensions. Now it has been shown that since you are progressively decreasing lambdas, you can actually get away with most of them. Typically, people take the first three values here, right? So the first 1, 2, 3. So now it's like a yi double prime. So you only take 3 values from this vector, and that brings you with dimension reduced to 3. So this is how for every element. So for this is for the ith element. Likewise, you can have for any element. So likewise, you would have it for all the elements, and now you can reduce it to a 3D space, and you can plot 1 and 2, 2 and 3, and 1 and 3. All right, so this brings us to an end of today's session. We don't have any practice problems for this session today. But yes, you have the assignment due day after tomorrow, Wednesday. This is your final assignment. Don't forget. And the exam is on Sunday. So before that, we would like to have a revision session. That's on Friday. You'll get an email notification as you get for the other sessions. So please attend this if you have any questions, and if you would like to have a quick recap of all the things. And as always, the PPT and the scripts will be uploaded to the Google Drive, and the recording of today's session will be uploaded on YouTube. All right. So I think, yeah, we are done with the session. Please let me know if you have any questions, anything to clarify. Otherwise, okay, we'll meet on Friday. All right. No questions? All right. Yeah. Thank you so much for attending the session. I'll see you all on Friday. All right. Bye-bye.